you see outside and inside Africa, you can estimate approximately when that happened. And it's sort of less than 100,000 years ago when this exodus out of Africa started. So that is sort of the genetic evidence for the recent African origin of modern humans that one sees. But there is then a problem with this idea, if you like, saying that modern humans evolve in Africa, come out and colonize the rest of the world. And that is what Jean-Jacques Heblin had talked to you about, that there are other forms of humans that exist, also in Eurasia, that go back almost 2 million years. And most well-known, of course, are more recent forms of humans in Western Eurasia, the Neanderthals, and in Eastern Eurasia, other forms that are less well known. So a big interest in our group have since 20 years been, what happened to these groups? Did they contribute genetically to people today? How are we related to them? But the problem is, of course, that they are extinct, so you will have to then try to retrieve DNA from old remains. And that work goes back over 30 years in the meantime, started out by studying ancient Egyptian mummies back in the 80s, where in some mummies you can see, for example, here in the skin and the basal levels of the skin of the 2,300-year-old mummy, structures that look like cell nuclei where DNA would be preserved. And you can also stain these for DNA, so there is DNA preserved. Back at that time, I then went on to clone that DNA and actually sequence it and show that there was human DNA preserved there. But we just thought it was preserved. In reality, what one didn't realize at that time was that this type of work is very prone to contamination. So most likely the DNA sequences I determined back then were DNA sequences from myself and not at all from the mummy. So the sequences you should not believe. Because what then have turned out is that, of course, in such old remains, there is very little DNA preserved, in the order of 100,000 or 1 million-fold less DNA per gram of tissue than they would have from fresh tissues. In addition, there are chemical modifications in it, and the vast majority of all the DNA in such a specimen doesn't come from the individual itself, but from bacteria and microorganisms that have colonized the tissues after death. And this then means that also small amounts of contamination of present-day DNA that may enter your experiments, that will not show up at all if you study present-day DNA, can sort of totally overwhelm your results here and dominate what you see. So this led then over 20 years to more and more sort of paranoia of cleanliness, where you start wearing funny clothes when you do your experiments, work in separate laboratories, sterilize all reagents and instruments you use, and so on. And it also resulted in that one focused one's work not on the nuclear genome, but on a tiny part of the genome localized outside the cell nucleus, the mitochondrial genome. It's a tiny piece of DNA. It's inherited only from mothers to offspring, so it reflects only the female side of history. And the advantage of it, though, is that it occurs in many copies per cell. So there are hundreds, sometimes thousands of copies of mitochondrial DNA per cell, whereas in the nuclear genome, there are just 2 copies, of course. The genome you inherited from your mother and from your father. So one have then used these techniques to go back in time, study zoological collections, more recently extinct animals that became extinct in the last 100 or 200 years, such as the marsupial wolf in Australia, animals that disappeared during the last glaciation, such as ground sloths or mammoths. But what is our big interest are then particularly the Neanderthals. So the Neanderthals then appear, I sort of hardly dare say anything here, but depending, I guess, on how you define a Neanderthal, maybe somewhere 300,000-400,000 years back in time and lived to somewhere between 30 or 40,000 years ago when they become extinct. And to the left you see a sort of reconstructed Neanderthal skeleton compared to a modern human skeleton. And there have been different ideas around what happened when modern humans then come out of Africa. One idea was a total replacement where Neanderthals become extinct in Europe with no mixture at all. Asian forms also, no mixture at all with modern humans. And the other idea is that yes, Neanderthals become extinct, but they contribute a bit to the genetic variation of Europeans today. And you can, of course, imagine anything from 0% contribution to quite a bit of contribution here. So the first chance then to test this with genetic means came in the mid-90s when we got access to the Neanderthal type specimen. So this is the specimen that was found in 1856 in Neanderthal in Western Germany and gave its name to this group of hominins. Looked at the sample from the humerus there, focused then on this mitochondrial DNA, a particular variable part of it, very cumbersomely with the techniques of the time, retrieved short pieces many times over, reconstructing a longer piece of DNA, believing the substitutions that you repeatedly saw there. And if you then estimate such a tree of the relationship of the mitochondrial DNA then with present-day humans, the picture was very clear. All present-day human mitochondrial DNAs go back to a common ancestor 100, 200,000 years ago. Much further back, 500,000 years or a bit more, is there a common ancestor shared also with the Neanderthal mitochondrial DNA. Since then, we and others have looked at many other Neanderthal mitochondrial DNAs, and this is a consistent picture. So it's very clear there are no humans today that run around with a mitochondrial DNA from a Neanderthal. So in this scheme then, it's total replacement. But it's also clear, as I have stressed, that the mitochondrial DNA is a very small part only of the genome, and that the big, complete picture would be in the nuclear genome. And the chance to sort of start at least dreaming about studying the nuclear genome of the Neanderthal really came in the early years of this millennium, when new techniques came around to sequence millions and billions of DNA molecules rapidly and quite inexpensively. So you could then start thinking about extracting the total DNA from such a specimen, sequencing all the DNA that's hiding in the bone, make a little data bank, and start comparing that data bank to the human genome that was now sequenced to see which molecules of DNA there might come from the Neanderthal. The first place where this worked was in Croatia, a beautiful site called Vindija, from this bone that may be around 40,000 years old. And we were then very lucky to get funding to improve over 5 years our techniques with which we extract the DNA from the bones and modify them to a sort of form that can be fed into these sequencing machines. So we got a lot better with that, the machines got better, and one of the things we then discovered was that the DNA that is similar to the human genome and therefore likely to come from the Neanderthal is indeed of very short size in there, 40, 50, 60 nucleotides long, whereas if you extract DNA from a blood sample from me, you will easily have things that are 10, 20,000 nucleotides long. The other thing that became obvious was that in most cases there's very little endogenous DNA from the Neanderthal there. Our very best bones have something like 3 or 4%. All the rest are then from bacteria and fungi that have grown in the specimen when it was deposited in the cave. We also found out other things, for example, that there are certain modifications in the DNA towards the ends of the molecules. We find very many apparent substitutions from C to T, unreasonably many. And it turned out to be due to chemical modification of one of these bases, C that lose this chemical group.